Hello everyone, just waiting for a few more people to come into the lesson space and then we'll start. Okay, let's go from here. So welcome everyone to the third session today, I think, and the last session of the My Tutor online school. So today is GCSE Biology. We base our GCSE biology lessons on the AQA specification, but we keep the topics very general. So if you're not studying AQA, then don't worry, it will be relevant. Today's lesson is on photosynthesis, and it'll be about 45 minutes of content and then about 15 minutes of questions. But what I usually do is have little breaks in between where I try and answer some questions about the topic that we've just covered. My name's Charlotte. I am the biology tutor for the My Tutor online school. Hi again. If you've tuned in before, if you haven't, then this is a good place to start because we're starting a new topic today. I have been tutoring with My Tutor for 2 or 3 years now. I studied conservation and biodiversity at the University of Exeter, and now I work for a charity helping farmers to do good things for wildlife and the environment. So I'm sort of an ecologist and a general biologist. So let's move on to today's slides. So last time we went through a revision of a cell biology topic. We did a little bit of mathsy stuff just to make sure that we were clear about that. And then we had a quiz and we did some past paper questions and cleared up any questions about parts of the cell, transport in cells, mitosis, and stem cells, growth and differentiation. So that's what we did last time. This time we are looking at photosynthesis. So we're starting a new topic. And this week, both biology sessions, we've got another one on Friday, are going to be on photosynthesis, going through that process. So what I'll do today is we've got a couple of different sections and then some mini quizzes and some past questions as well. The way that we tend to run sessions is that if you look at the bottom of your screen, you've got a chat button and a Q&A button. Any answers to mini quizzes or anything like that can go in the chat, and any questions about the content, if you pop them in the Q&A. Now I'll try and keep things a bit cleaner and make sure that I don't miss any questions. Okay, so I thought I would start with a word cloud to find out what you already know about photosynthesis. Kind of get us all moving because I know it's after lunch and I always feel a bit sleepy after I eat. So let's go to Slido. So what I'm going to do is I'm going to activate this word cloud and I'm going to share the link in the chat. So if you go to the chat, then you should see a link now to Slido. And if you click that link, it will take you to a screen in your browser where you can answer the question, what keywords do you know about photosynthesis? And it will populate a word cloud, which hopefully you should be able to see any second. Here we go. Okay, chlorophyll, plants, respiration, sunlight, light. Good, loads of good keywords so far. What else can you think of about carbon dioxide, glucose? Good. Oxygen, seaweed. That's a good good observation. Seaweed and photosynthesis, yeah. Energy, light, water, carbon dioxide, green. Yes, green is definitely a key word. Loads of people putting in water, that's great. Sunlight, photosynthesis in general. H2O, required practical. Good. Fab. Stomata, yes, really good. Great. Substances, thylakoid discs. Gosh, that's technical. That one comes in at A-level. Oh, C6H12O6. Yes, that is the chemical formula of glucose. Well done. Loads of stuff in here. Xylem in here as well. Substances, roots. Brilliant. Loads of good stuff here. Okay, let's stop that one then. And move on to what we know. So all of those keywords that you put down in that word cloud today, we should cover today, apart from possibly thylakoid discs, but I will mention them. So that's kind of a good start to today, a good baseline of keywords, things that you need to look out for today and to remember. So I thought we'd start with kind of what photosynthesis is and why plants do it. So plants don't need food for respiration, growth, or reproduction. They produce their own food by the process of photosynthesis. So whereas we eat food in order for us to grow, to move, to do all of the things that we do with our lives, plants produce their own food by this process of photosynthesis. And we've got a really nice diagram here of well, kind of the whole process of photosynthesis. So I didn't want to go straight into an equation, but I thought this was quite fun. So we've got water down here that's absorbed from the roots, carbon dioxide that enters through the stomata of the leaves. A couple of people put down stomata on that word cloud. So here's stomata are where gas exchange happens in leaves. So they're those gaps in the underside of a leaf. Carbon dioxide enters through those and is then used in photosynthesis. At the same time, oxygen goes out. We'll come to that later. Light from the sun also shining on the leaf, and then within the leaf we have chloroplasts, those plant organelles that are present in most plant cells. They're filled with a green pigment called chlorophyll. You can see here, those of you popped that in the word cloud as well. And the chlorophyll traps light energy to make food for the plant. So all of these three things come together, and then in the leaf, the process begins. And in that process, the water and the carbon dioxide are changed into oxygen, which you can vaguely see on this arrow here, which is given off into the air via the stomata we talked about just now, and also sugar. And the sugar is in the form of glucose, which we've talked about before when we talked about digestion. Glucose is a really, really useful product. You can convert it into almost anything. So that sugar in this diagram, they've said, is converted to starch, which is how plants store food. So they store their food as starch, and it turns iodine a dark blue. So this is kind of an overview of photosynthesis. We're going to go into it all in a bit more detail. I thought this was quite a nice diagram. I like the colours, I find it easy to remember. So here's the photosynthesis equation. So carbon dioxide plus water with light on the arrow there gives glucose and oxygen. This hasn't transferred quite as well from my PowerPoint slides as I'd hoped, so it should all be in one line rather than stacked on top of each other. But this is the question that this is the equation that you need to remember for photosynthesis. Carbon dioxide and water with light produces glucose and oxygen. In chemical form, CO2 plus H2O gives C6H12O6, which is the chemical formula for glucose, plus O2. this equation needs to be balanced, and you might well be asked to balance it as part of the exam. It's a fairly easy one to balance, so you shouldn't have too much trouble. But to balance it, we need to make sure that there's the same number of molecules of each, or atoms of each element on each side of the equation. So I find it easiest to start with the glucose here. You can see that there's 6 carbons here on this side of the equation. So on the right-hand side of the equation, and on the left-hand side, we only have 1 at the moment. So let's start by balancing that out. And let's put 6 here. So 6 CO2 plus H2O gives C6H12O6 plus O2. Okay, so we've done the carbon. The next thing that we can do is the hydrogen. So we can see that there's 12 hydrogens in the glucose. And on the left-hand side here, we have 2 so far. So there are 2 molecules, 2 atoms of hydrogen in every water molecule. 2 fits into 12 6 times. So we could also balance our H2O by adding a 6 in front of it. This just leaves the oxygen to balance. So at the moment, you have 6 oxygens in the glucose and 2 oxygens in the oxygen itself, so 8 altogether. Whereas on this side, we have 12 in the CO2, so O2 times 6, 12 oxygens, and then another 6 here, so 18 altogether. We've got 18 oxygens here. We need to balance this, and the easiest way to do that is to put a 6 in front of this oxygen as well. We have 12 here and another 6 here to make up to 80. So that's how we balance the equation here. Okay, you see a few people are having issues with the audio, so not being able to see or to hear. Frederica, if you're there, can you give me an idea of whether you can see or hear me?
All good from me. I can see you and I can hear you. If there are people struggling with audio and not being able to see, then something that you could try is maybe relaunching the link in an incognito window. But all good from me.
Perfect. Okay, I'll carry on then. I can see a couple of you have kindly popped in the chat as well that you can see and hear me, so at least I know it's not for everyone. Okay, so we've balanced the equation for photosynthesis. So we know that 6 CO2 molecules and 6 water molecules are converted to 1 glucose and 6 oxygen molecules. So we had to work quite hard to get that 1 molecule of glucose. It needs quite a lot of different things. This reaction is an endothermic reaction. That means that it needs an input of energy from the environment. So the energy transferred from the environment when the bonds holding carbon dioxide and water are broken is more than that transferred back to the environment with the formation of the new bonds in glucose and oxygen. Endothermic needs help, needs more energy. And we get that from the light. That's why the light comes in there as well. So you need to know that photosynthesis is an endothermic reaction. Requires energy. Okay. So photosynthesis equation might look familiar to a few people because it is the same components as respiration, but backwards. So we're going to look at respiration next week, so don't worry if you haven't heard about respiration or you need a refresh. But photosynthesis is essentially all the same components, but going the other way around. So in respiration, glucose and oxygen are converted to carbon dioxide and water. So in reverse. So it's quite cool, really, because plants effectively kind of have almost like a recycling mechanism. So they're using photosynthesis to make their food, and then respiration of that food provides them with the energy and the other things that they need to survive. So they have this whole kind of cycle of photosynthesis and respiration going on. How are plants adapted for photosynthesis? There are lots of ways that plants are adapted for photosynthesis. So I really like this diagram here because it shows you the kind of just what I was talking about there, that there's lots of photosynthesis which is happening during the day when there's light available. But then there's also respiration going on, which is happening during the day and at night. And you can see on here, I hope you can see anyway, that for photosynthesis, you've got carbon dioxide coming in and oxygen going out here, all through the stomata on the leaves. And they've also popped water in there and carbohydrates coming out. And by carbohydrate in this diagram, they mean that glucose. Then they've also at the bottom got respiration. So they've got oxygen going in this case, through stomata at night, and then going out. They've got carbon dioxide and water. Energy being produced there as well from respiration. Plants are really well adapted for having both of these processes going on and making sure that they can do enough photosynthesis to justify the respiration they need to do. So leaves are very broad. They have a really big surface area for light to fall on. We have talked about having a big surface area, a short distance, and different concentrations over and over again so far in these tutorials. It's really important. We talked about it particularly with diffusion and osmosis as well, and gas exchange and then digestion and all of those other things. So here again in photosynthesis, we have similar factors coming into play. Having a big surface area means you can have lots of chloroplasts facing the top of the leaf, and you can get lots of sunlight for photosynthesis. Leaves are very thin, which means that the diffusion distances for gases are short. So I said that carbon dioxide and oxygen are going in and out of the stomata, and water vapor is going in and out of there as well. So they need that short diffusion distance for that to happen quickly. Plants have chlorophyll. That is a major way that they are adapted for photosynthesis. So the chlorophyll is the pigment in the chloroplast that allows them to absorb the light. Leaves have veins. So veins are kind of like the roads in the leaf. So they bring water in the xylem to the cells in the leaves, and they remove the products of photosynthesis in the phloem. So within the veins, you have both xylem and phloem cells, and we've talked about xylem and phloem before. So xylem are how water is transported, and phloem are how kind of nutrients and particularly glucose and things like that are transported in the plant. So the veins are how the plant allows those transport systems to happen. Air spaces in leaves. So they allow the CO2 to get to cells and O2 to leave by diffusion. In the case of photosynthesis, talking about respiration, it's the other way around. So you can see in this diagram here, which is of kind of a cross section of a leaf, got the waxy cuticle and the upper epidermis at the top here. Then we have our palisade layer with loads and loads of chloroplasts. This is the top layer, so that's where the sunlight's hitting. And then we have this spongy mesophyll layer. Got a vein there, and you can see the two different coloured dots there. They represent the xylem and the phloem, so that's the transport system. The spongy mesophyll layer, you can see the cells aren't packed so closely together. There's lots of space around them for the gases to diffuse in and out. That's really key. And then you can see the stomata and the guard cells at the bottom there, that gap that allows the exchange of gases, and also water evaporates through there as well. Guard cells we've just kind of talked about, so they open and close stomata to regulate gas exchange. So guard cells are kind of like little curved cells either side of a stomata, and at night they close. And then they would open up during the day. And it means at night the plant can keep water in, basically, so it doesn't dry out. Okay. While we're here, we should probably mention those stacked thylakoids that someone put in the word count. So plant cells, as we've said, are adapted for photosynthesis with chloroplasts and chlorophyll. That chlorophyll within the cell itself, within the chloroplast, and you learn more about this at A level, so you don't need to kind of retain this little bit of information if you're not sure. But it is interesting. So when you're in a chloroplast, there are these stacks of grana and thylakoids. Thylakoids are like little kind of discs of chlorophyll. They're all stacked on top of each other. So that's how the chlorophyll is arranged within the chloroplast. As I say, you'd learn more about that at A level. Okay, mini quiz. So today we're just going to do this in the chat because last week we had some people could do some different kind of techniques that we tried and some people couldn't. So we're just going to do this in the chat. So what I'll do is I'll read out the question and then you pop in the chat A, B, or C. So question 1, what happens to glucose produced by photosynthesis? A, it's used in respiration and stored as starch. B, it's used in respiration and stored as phloem. Or C, it's transported in the xylem. Question 1, A, B, or C. Pop it in the chat. I'll give you 10 seconds. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Let's have a look. The correct answer to number 1 was A. Well done, everyone. Brilliant. So glucose is used in respiration and stored as starch. It's not stored as phloem. Phloem is the type of cell where stuff is transported, and it's not transported in the xylem. It's transported in the phloem. Question 2, how many molecules of oxygen are produced for each molecule of glucose in photosynthesis? So thinking back to that balanced equation, 3, 6, or 9, A, B, or C. A is 3, B is 6, C is 9. Pop the answer in the chat. Five, 4, 3, 2, 1. Good, loads of people putting B. Well done, everyone who put B. There's 6. Magic word when you're balancing the photosynthesis equation. 6 of everything except glucose. Number 3, photosynthesis is A, exothermic reaction, B, endothermic reaction. A or B? Pop your answers in the chat. Good, loads of people putting B. So it is an endothermic reaction. So it takes energy from the environment to make that reaction happen. Okay, if you've got any questions about the last few slides, pop them in the question and answer. I'm going to have a quick whiz through now. Have a look. Okay, someone asking what xylem. So xylem are cells in plants that transport water. They're pretty cool because they're technically dead. They don't have a nucleus. When they first form, they form a really tough structure around their outside from a substance called lignin. And that lignin solidifies and then forms that kind of really hard shell around a xylem vessel. It makes them perfect for transporting water because they're basically just a straw. They don't have anything else in them to get in the way. They just have these lignin sides. So they're almost like lignin tubes. And they're kind of, so quite often, obviously the stems of plants tend to be quite tough, and that's because they have lots of xylem vessels in them. It's that lignin that makes them feel quite tough. So xylem are how water gets around a plant. So they'll be in the stems and the veins on the leaves as well. Yeah, someone's just put in the chat. There have been a couple of issues with Zoom over the last few days by the sound of it. This is the first time I've used it in a couple of days, and so far it seems to be working okay with me. But it seems that some of you might be having difficulties. So try all of those things that Frederica suggested, and hopefully they'll get it sorted out in the next few days. Okay, let's move on. So the next things that we're going to concentrate on are what affects the rate of photosynthesis. So photosynthesis, we've learned, is a really good reaction. It produces lots of food for the plant, and we want to do it as much as we possibly can. So what are the things that affect photosynthesis and allow the plant to manipulate that speed of that reaction? So the first thing is light. And we call light a limiting factor of photosynthesis. So it's a factor that can mean that the reaction is limited. So the amount of light directly influences the rate of photosynthesis. We've got a nice graph here. You can see we've got light intensity on the x-axis, so going from small to large on the x-axis, and then we've got the rate of photosynthesis on the y-axis. And then we've got 3 letters on here, and I've written kind of a little explanation of what's happening at each of those letters. So at A, there's very little light intensity, and photosynthesis barely takes place, or it stops altogether. You can see it's very low there. But at B, we look at the graph there, B comes at a greater light intensity and also a greater rate of photosynthesis. So in general, between A and B, we have this fairly straight line, which indicates that as light intensity increases, the rate of photosynthesis increases as well. Positive correlation here. But then at B, or just after B, we start to get a flattening out of the curve. So just after B, it doesn't matter how much you increase the light intensity, the rate of photosynthesis stays the same. It doesn't drop, it just stays the same. So when the optimum light intensity is reached, the graph plateaus. So that's what we say when a graph flattens off, it plateaus. Light intensity is now no longer the limiting factor. Temperature or carbon dioxide concentration are now limiting the rate of photosynthesis. So we're going to look at temperature and carbon dioxide next, but those 3 kind of work together or can combine to affect the rate of photosynthesis. So when this graph plateaus, it means that light intensity is the best that it can possibly be, but there's something else stopping that rate of photosynthesis from increasing anymore. Okay, so here's the graph for temperature. And again, we've got the same axes on the graph. We've got the rate of photosynthesis on the y-axis, and we've got increasing temperature on the x-axis. This graph has some really nice explanations on it. This is exactly what you need to know. So to start with, we've got this kind of upwards curve. So as temperature increases, rate of photosynthesis increases rapidly, because it's a curve. So increasing molecular collisions due to increased kinetic energy leads to an increase in the rate of photosynthesis. So more temperature, more movement, everything happens faster. Then we reach a peak. So this peak generally happens at 35 to 40 degrees C. This is the maximum rate of photosynthesis. So you're now at the optimal temperature, where the rate of photosynthesis is the highest it's ever going to be. The reaction is happening very efficiently. It's very fast. This tends to be at 35 to 40 degrees C, but we're talking about enzymes. So enzymes are working to make this reaction happen. There are biological catalysts, and enzymes, we know, each have their own optimum temperature. Now in plants, this is generally 35 to 40 degrees C, but of course, plants that are adapted to different climates may have different optimum temperatures. If they're adapted to somewhere very hot, their enzymes might function really well at, say, 40 to 45 degrees C. After the optimum temperature has been reached for an enzyme, if the temperature increases again, something happens to enzymes, and there's a particular word for it. It begins with T. If anyone knows it already, pop it in the chat. Good, yeah, a couple of people popped that in there. It is denature. So after the optimum temperature, the increased movement and temperature basically disrupts the structure of the enzyme. And if you remember, enzymes are proteins, so they're made up of tightly coiled and arranged structures of amino acids. And enzymes in particular have a particular structure because they need to be complementary to their substrate. So if the bonds in an enzyme structure break and they start to lose their very specific shape, they can't bind to their substrate, and they're described as being denatured. They can no longer perform their function. This means that the rate of photosynthesis decreases because the enzyme can't bind to the substrate and the reaction can't happen. Generally, this is between 40 and 50 degrees C, but as I said, different plants are adapted to different temperatures. So you might find that in an exam, they'll give you a different example. And finally, carbon dioxide concentration. So this looks very similar to the light intensity graph, and that is because it basically has exactly the same shape. So we're looking at the same things again. X-axis is carbon dioxide concentration, y-axis is rate of photosynthesis. And again at A, at very low carbon dioxide concentrations, photosynthesis barely takes place or stops altogether. But then between A and B, we have this rise. So this, what you would describe as a positive correlation on the graph. As carbon dioxide concentration increases, rate of photosynthesis also increases. So at B, we're approaching kind of the maximum carbon dioxide concentration where rate of photosynthesis is still increasing on the graph. But then when we get to C, the graph has plateaued, so it's leveled out. And carbon dioxide concentration is now no longer the limiting factor. At this point, temperature or light intensity are now limiting the rate of photosynthesis. So that leveling out, because it doesn't dip, so that temperature, the graph dips. And that's because there's something directly happening there, the denaturing of the enzymes. Here, it's not affecting the way that the reaction can work, it's just that something else isn't high enough to keep increasing that rate of photosynthesis. So here's all of those graphs together. So it's quite often how you'll see them in revision guides and things, and it's good to be able to see them all together. So here you can see they put some optimal light intensities, temperature, and carbon dioxide concentrations on there as well. So light intensity on the left here, low light conditions decreases the rate, whereas increasing light intensity past the optimum light intensity has no effect on the rate of photosynthesis. It's a very posh way of saying that it increases until it levels out. Temperature, you can see here, is just an up and down. Again, it increases to that optimum temperature, but then after that optimum temperature, enzymes start to denature and the rate decreases. Finally, carbon dioxide concentration, which is the same as light intensity. So you get an increase up to a point where the graph levels out. They reckon that that optimum carbon dioxide concentration is about 0.1%. It's not very high. 0.1%, one carbon dioxide in every 100 molecules, every 1,000 molecules. Remember, so these three factors all work together to determine the rate of photosynthesis in a plant. But that assumes that the plant has lots of chlorophyll to start with in order for light to be absorbed so that photosynthesis can take place with the energy that it needs. So the amount of chlorophyll in a leaf also controls how much photosynthesis can take place. So it's really important to maintain that chlorophyll. And you can do that by some of the adaptations that we mentioned earlier. So leaves having a really great big surface area, having all of those palisade cells with lots of chloroplasts at the top of the leaf so they get the sunlight. And then also by making sure that the plant is healthy, that it has enough minerals like magnesium to help maintain photosynthesis rates. And we have talked about minerals in plants before. We talked about nitrate and magnesium and how they affect plant growth. Magnesium is a mineral that's found in the soil and it is absolutely essential for making chlorophyll. So without chlorophyll, obviously the plant can't photosynthesize and growth is stunted. So magnesium is really essential to the plant for producing and maintaining that chlorophyll. Measuring the rate of photosynthesis. Required practical. Some of you might have done this in the lab. It's a lot more fun to do in the lab than watching it on the screen here, so I'm sorry about that, but this is the best that I can do. If you haven't done this before, it is quite a fun experiment and you should get a chance to do it next year. Hopefully, we hope. It sounds as though you will. So basic setup is light, a way of measuring how far that light is from the plant, and then the plant itself is a water-going plant. And the water plant is in some water, and there's a test tube over the top of that container that allows you to collect or measure the bubbles of oxygen gas that are being emitted from that plant. Why do we use a water plant? So that we can see the gas that's being given off. So that oxygen gas is evidence of photosynthesis happening. That is a waste product of photosynthesis. But normally we can't see the bubbles of oxygen that plants are producing because they're out in air. Using a water plant allows us to see that evidence. And you can see on this kind of table here in the graph. So let's start with the table. Distance between the pondweed and the light source in meters. So they've started with the light source quite far away and they've moved it closer each time. So for this experiment, probably our hypothesis is going to be that as the light source gets closer to the plants, the rate of photosynthesis is going to increase. And therefore, we should see an increase in the amount of bubbles given off. They've moved it closer. They've also measured the light intensity, so they've done power divided by area of light intensity, so watts per meter cubed. They've measured that. And we can see that the light intensity does increase as we get nearer to the plant, which is what we would expect. Light source gets nearer, there's going to be more light landing on more of the plant. Number of bubbles given off in 1 minute. That's the right-hand side of this table. So you can see that goes from 8 to 28 to 105 and then remains at 105. And they've plotted those results on this graph. The number of bubbles given off in 1 minute is on the right-hand side, light intensity on the x-axis at the bottom. We can see that rise in number of bubbles given off as light intensity increases, and then it plateaus, so it levels off. And that's when, rather than light intensity being a limiting factor, temperature or carbon dioxide are instead. Okay. Mini quiz. Okay, so ready on the chat. Mini quiz. Number 1. If temperature increases beyond the optimum temperature, what may happen to enzymes involved in photosynthesis? Some of you have already answered this in the chat, but if you could pop it again, that would be great. So we're looking for a word here. What happens to enzymes involved in photosynthesis when temperature goes beyond the optimum temperature? Good, loads of people putting the right answers in the chat, well done. Enzymes do denature. There's a couple of people putting the enzymes die. So they kind of do, but I see it in mark schemes a lot, don't accept die. So enzymes denature, but they don't die. That's the sort of thing that might just miss you 1 mark, and it's worth every mark you can get when you get into that exam. So just be really careful there with language. It's one of those things that's a bit silly because they kind of do die, but technically they don't. So denature is the way to go. Question 2, which limiting factor does not plateau at an optimum level? A, light intensity, B, temperature, or C, carbon dioxide concentration? A, B, or C in the chat. Good, again, loads of people putting the right answer in here. Keep going. Keep popping your answers in the chat. Good. B was correct. So temperature doesn't plateau. Temperature does that up and then down. So 3, other than temperature, carbon dioxide concentration and light intensity, what other factor may affect the rate of photosynthesis? Pop your answer in the chat. Not temperature, not carbon dioxide concentration, and not light intensity. What was the other thing that we had to remember affected the rate of photosynthesis, or whether photosynthesis could happen at all? Good. Few different answers coming in here. They're all right so far. Well done. Not temperature, no. Availability of water, that's really good actually. We haven't really talked about that, but yes, water is needed for the plant to survive in the first place. But don't forget that respiration going on there as well. Chlorophyll was what I was looking for, or nutrients. Both of those have come up. So chlorophyll obviously is needed to absorb the light energy. Nutrients from the soil might be needed to maintain that chlorophyll. And we talked about one nutrient in particular. We talked about magnesium. Good. Okay. I'll just have a quick whiz through some questions, and then we've got 10 minutes or so to do some exam questions. Why do some plants under sunlight become yellow? That's a really good question. So, yellow plants can be yellow for lots of different reasons. So, one of those reasons that's quite likely is that they haven't got enough nutrients to maintain that chlorophyll, and that's where that magnesium comes in. So magnesium is directly associated with chlorophyll and creating that and maintaining it. Another nutrient that's really important is nitrate, and we're going to talk about that a bit more on Friday, but nitrate is really important for growth of the plant. Nitrate is particularly important for producing proteins, and proteins are what allow reactions to take place in a plant. Proteins are also enzymes, so magnesium and nitrate are particularly important. Other reasons that a plant can become yellow, some diseases make plants go yellow as well. So right at the beginning when we talked about pathogens and disease, which was a very apt topic for the beginning of our online school, we talked about how plants can become yellow if they're infected by different diseases. So tobacco mosaic virus is one of those, produces those yellow spots. Does a lack of nitrate ions result in stunted growth? Yes, exactly. Without nitrate, we can't produce proteins, and without proteins, we can't do those reactions to grow. So yes, nitrate ions result in stunted growth. Interestingly, magnesium does too, but for a different reason, because without magnesium, we don't have healthy chlorophyll. Without chlorophyll, we can't complete photosynthesis, and again, can't grow properly. So the first symptom often of a nutrient deficiency is a lack of growth. If there's too much light, would it not burn the planet? Oh, burn the plant. Yes. Usually, when you're doing a lab experiment like that, there's not really a danger of that. But sometimes you can use a heat shield as well. And in some schools, they do use those as well. They make sure they have a heat shield to try and keep the temperature the same, if nothing else, because in an experiment like that, you need the temperature to be the same for it to be fair. And you're quite right, if you get too much light intensity, the temperature's like to be really high and the plant might get scalded. Please could I go back to the slide with all 3 graphs about chlorophyll? Yes. There you go. Lots of people mentioning the lock and key theory. Yeah, that's exactly right for enzymes. So lock and key is how they form those enzyme substrate complexes. Do you know any methods to kill tiny spiders on roses? Sort of. So depends what you mean by tiny spiders. If they are tiny spiders, they're probably not doing the rose any harm. If they're aphids, then aphids can be a problem on plants, and then the best solution is ladybirds. So if you can encourage ladybirds into your garden, get loads of nice different plants, then they can be really helpful. You can also use pesticides, but I'd recommend them as being a last resort because they also kill all of the good stuff as well. We don't want that. You just want to kill the things that are affecting the plant. So if you can, it's best to allow nature to take its course and introduce the predators of those things. So if they're aphids, then ladybirds are the way forward. Okay, let's do some past exam questions. Okay, so last time I had a few people saying, oh, can we do these in the chat before you reveal the answers, or just write them down on paper if you have a pen and paper in front of you, and then you can check them as I reveal the answers. So I have done that this time. So we've started with a really simple question. Let's underline the key words and check the marks. So complete the word equation for photosynthesis. Remember, in an exam, you're going to need to underline photosynthesis because it's very similar, the equation, to respiration. You want to make sure you get it the right way around. Remember, in an exam, you might be slightly stressed, you might be in a hurry, you've got to the last 20 minutes, you have more questions than you thought to go, and you don't want to get this one wrong. You need to use the words from the box, and it's 2 marks, so 1 for each. Okay, well done those who've had a go. You have all got it right so far. Okay, yep. So the answer to this one is carbon dioxide plus water gives glucose and oxygen. The way I kind of remember it is that, so we need oxygen. We are not plants. Plants need the carbon dioxide that we breathe out to do their photosynthesis. So that's how I remember it. So plants need the stuff that we breathe out. So the equation starts with carbon dioxide, but that's a slightly kind of backwards way of remembering it. So I understand if you'd prefer something else. That's how I think of it. Okay, this is a experimental question. So it's a bit small, but I'll read it out. Hopefully you can see it. I had to try and get it all on 1 page. So plants may grow faster if they have more carbon dioxide. Indigestion tablets dissolve in water to form a solution, and this solution slowly gives off carbon dioxide. A student sets up an investigation to see what concentration of carbon dioxide is best for increasing the growth of a geranium plant. The student put a geranium plant in a clear plastic bag, which you can see, put a dish containing water and 1 tablet in the bag, and then sealed the top of the bag. So you've got your plant and your dish with a dissolving tablet in it, also in the bag. The students set up 5 more experiments, each with water and a different number of tablets. Then they left all the plants in a well-lit place for 4 weeks. This was a long experiment. The student used a clear plastic bag, not a black plastic bag. Explain why. This is an explain question, so they want to know about the biology behind something. It isn't describe, it's an explain question, and it's 2 marks, so we need to make 2 points. Have a little think why a clear plastic bag might be better than a black plastic bag. Here is the answer. Clear plastic bag would let light in so that photosynthesis can take place to produce glucose. You've got 1 mark here for saying that a clear plastic bag would let light in, and you've got the other mark for giving the kind of more biology behind that. So linking that light to photosynthesis taking place and that photosynthesis is needed to produce glucose. Okay. So well done there. I also felt, and they didn't really have it in the mark scheme, but surely a black plastic bag, it would get real warm in there. And you don't really want to be overly influencing the temperature when you're really supposed to be looking at carbon dioxide. Okay, here we go again. After 4 weeks, the student counted the number of new leaves on each plant. The graph shows the results. So we've got 2, 4, or 6 leaves and 1, 2, 3, 4, 5 tablets dissolved in water. Describe the effect of increasing the number of tablets dissolved in water on the number of new leaves that grew in 4 weeks. This is describe here. They don't want it explained, they want it described. They've given you 3 marks for it. So 3 marks for describing that graph. When you're looking at a graph, make sure that you look at the units, so number of tablets we looked at and number of leaves. Make sure you're clear on the axes and make sure you look at this pattern on the graph. So to start with, as number of tablets dissolved in water increased, number of new leaves after 4 weeks increased. Then what happens? Then it plateaus over. So let's look at the worked answer. To begin with, number of new leaves increases with number of dissolved tablets. Then after 3 tablets, the number of new leaves stays the same, about 5 new leaves after 4 weeks. That was all that you needed for 3 marks. And given that the axes are so long, it would take quite a long time to write that. Just takes a long time just to write number of tablets dissolved in water increases with number of new leaves after 4 weeks. So no wonder they gave you a bit of space. What you also needed for this question, because it was a described question, was to link it to the graph itself. So notice that in that answer, we've talked about 3 tablets and 5 new leaves. So we've directly taken those from the graph, that point there. So when you've got a described question like that, it's well worth referring to the specific numbers in the table or on the graph. Okay, I think we're going to leave it there for this week. I'm sorry that some of you were having technical problems. I hope that Zoom sorted out soon. I noticed that there was a few more than normal last week as well, so it sounds like it's been going on for a while. But fingers crossed, they will get that sorted out. And hopefully that'll be better tomorrow. We have another lesson on Friday. So on Friday, I will be going through how plants use glucose and going into the biology of that a bit more. And then also talking about photosynthesis and agriculture, how we can manipulate it in there. So that'll be Friday. So until then, have a good week. Remember that my tutor offers other things as well. So if you want one-to-one sessions or small group sessions, my tutor are putting those on, so have a look on the website. Double check the online timetable for the rest of the classes this week and make sure that you have some good evenings and go out in the sunshine. Thanks for coming today and hopefully see you on Friday.